Alright, so today is the cell cycle. I'm just going to read it. I'm preparing for an exam. And sometimes just getting on stream helps me study. Let me get my notes out. We got about 2 hours and 15 minutes to go over 20 pages. Seems doable. Oh crap. Oh boy. I'm gonna fudge this up. Okay. Alright. Chapter 12. Cell cycle. Textbook notes. Most cell division results in genetically identical. Cell division plays several important roles in life. When a prokaryotic cell divides, it is actually reproducing because the process gives rise to a new organism. The same is true of any unicellular eukaryote, such as an amoeba. As for multicellular eukaryotes, cell division enables each of these organisms to develop from a single cell, the fertilized egg. A two-celled embryo, the first stage in this process, is shown. And cell division continues to function in renewal and repair in fully grown multicellular eukaryotes, replacing cells that die from accidents or normal wear and tear. For example, dividing cells in your bone marrow continuously make new blood cells. The reproduction of a cell, with all of its complexity, cannot occur by a mere pinching in half. A cell is not like a soap bubble that simply enlarges and splits into two. In both prokaryotes and eukaryotes, a crucial function of most cell divisions is the distribution of identical genetic material to two daughter cells. The exception is meiosis, the special type of eukaryotic cell division that produces sperm and eggs. What is most remarkable about cell division is the accuracy with which the DNA is passed from one generation of cells to the next. A dividing cell replicates its DNA distributes the two copies to opposite ends of the cell, and then splits into daughter cells. Cellular organization of the genetic material. A cell's DNA, its genetic information, is called its genome. Although a prokaryotic genome is often a single DNA molecule, eukaryotic genomes usually consist of a number of DNA molecules. The overall length of DNA in a eukaryotic cell is enormous. A typical human cell, for example, has about 2 meters of DNA. A length about 250,000 times greater than the cell's diameter. Before the cell can divide to form genetically identical daughter cells, all of this DNA must be copied or replicated, and then the two copies must be separated so that each daughter cell ends up with a complete genome. The replication and distribution of so much DNA are manageable because the DNA molecules are packaged into structures called chromosomes. Speaking of, do I have my... And we don't need to do it. Each eukaryotic chromosome consists of one very long linear DNA molecule associated with many proteins. The DNA molecule carries several hundred to a few thousand genes, the units of information that specify an organism's inherited traits. The associated proteins maintain the structure of the chromosome and help control the activity of the genes. Together, the entire complex of DNA and proteins that is the building material of chromosomes is referred. Okay. Every eukaryotic species has a characteristic number of chromosomes in each cell's nucleus. For example, the nuclei of human somatic cells contain 46 chromosomes, made up of two sets of 23. Reproductive cells, or gametes, such as sperm and eggs, have half as many chromosomes as somatic cells. The number of chromosomes in somatic cells varies widely among species. 18 in cabbage, 48 in chimpanzees, 56 in... Okay, wow. When a cell is not dividing, even as it replicates its DNA in preparation for cell division, when a cell is not dividing, even as it replicates its DNA in preparation for a cell division, each chromosome is in the form of a long, thin chromatin fiber. After DNA replication, however, the chromosomes condense as a part of cell division. Each chromatin fiber becomes densely coiled and folded, making the chromosomes much shorter and so thick that we can see them with a light microscope. Each duplicated chromosome consists of two sister chromatids, which are joined copies of the original chromosome. The two chromatids, each containing an identical DNA molecule, they're often attached along their lengths by protein complements called cohesins. This attachment is known as sister chromatid cohesion. Each sister chromatid has a centromere, a region made up of repetitive sequences in the chromosomal DNA where the chromatid is attached most closely to its sister chromatid. This attachment is mediated by proteins that recognize and bind to the centromeric DNA. And other bound proteins condense the DNA, giving duplicated chromosome a narrow waist. The portion of a chromatid to either side of the centromere is referred to as an arm. An unduplicated chromosome has a single centromere distinguished by the proteins that bind there and two arms. You know, one thing that's pretty interesting is that the centromere is a region of repetitive sequences. That's pretty interesting. So later in the cell division process, the two sister chromatids of each duplicated chromosome separate and move into two new nuclei, one forming at the end of each end of the cell. Once the sister chromatids separate, they're no longer called sister chromatids, but they're considered individual chromosomes. This is the step that essentially doubles the number of chromosomes during cell division. Thus, each nucleus receives a collection of chromosomes identical to that of the parent cell. Mitosis, the division of the genetic material in the nucleus. Is usually followed immediately by cytokinesis, the division of the cytoplasm. One cell has become two, each the genetic equivalent of the parent cell. From a fertilized egg, mitosis and cytokinesis produce the 37 trillion... Wow, that's insane. That's freaking crazy, man. That make up your body. And the same processes continue... Hold on. In contrast, you produce gametes, eggs. Okay, so the answer to this is that there's one chromosome, now there's two chromosomes. All right. The mitotic phase alternates with interphase. Actually, you know what? I kind of want to do this. Browse, add. What we need to do is we need to look for some. A threading process. Kind of interesting. I forgot to write this down. We're not doing 16? 16 is probably a 6B thing then. Yeah. Oh, we're gonna do freaking protein electrophoresis? That's cool. Oh, we're gonna do a mini prep? That's fun. Oh, we're gonna do P-Glow? Wow, that's great. Oh, and then we're gonna do PCR? Oh, that's pretty cool. Why do we start with Dude, I'm so excited. What about the labs? I feel like the labs in this class are not gonna be like, field day? That's pretty rad. Dude, I'm so excited for 6B. Alright, so, but we're not doing 16. Wait, what the hell is the nucleolus? Each duplicated chromosome appears as two identical sister chromatids joined at their centromeres. Kinetic. Okay, wait. The nuclear envelope fragments, the microtubules extending from each centrosome, can now invade the nuclear area. The chromosomes have become even more condensed. A kinetochore, a specialized protein structure, has now formed at the centromere of each chromatid. centromere of each chromatid. Okay. Many of the events of mitosis depend on the mitotic spindle, which begins to form in the cytoplasm during prophase. Okay, let me just try to go through it. So prophase, it starts to condense, right? Is that right? Yeah, prophase, it's condensed. Prometaphase, I guess that's the main thing. The nuclear envelope fragments. Then in metaphase, they line up, and then anaphase, they separate, and then telophase, the nuclei start to form. Tubulin, dude. Wow, that's an amazing way to do this. They play tug of war until they even out. That's so cool. What an ingenious solution. I don't know why they don't call it the metaphase plane. Separates, bro. That's cool. Separates. In a dividing animal cell, the non-kinetic or microtubules are responsible for elongating the whole cell. How do they do that? Non-kinetic or microtubules from opposite poles overlap each other extensively during metaphase. During anaphase, the region of overlap is reduced as motor proteins attached Do they have compressive strength, these tubulin subunits? Because it makes sense to me that they work under tension because they can pull the freaking chromosomes, but do they work in compression? Wow, bro. Someone actually computed the Young's modulus? That's crazy. Bro, it's gassing me up, man. That actually worked on me. Yeah, I'm a freaking mechanistic cell biophysicist. Oh, what do you do? I'm just a mechanistic cell biophysicist. I don't understand. It's still getting pushed. I don't know if that was that satisfying. I'll be back. Right, cytokinesis. Let's do it. In animal cells, cytokinesis occurs by a process known as cleavage. The first sign of cleavage is the appearance of a cleavage furrow, a shallow groove in the cell surface near the old metaphase plate. On the cytoplasmic side of the furrow is a contractile ring of actin filaments. Oh, it gets chewed up like this way? That's cool. So it doesn't get wound around. I was imagining it's like you're pulling, like you're winding up a spool of yarn or something. Or not yarn, but like something like that. Tubulin, bro. Alright. But let's get back to business. Yeah. The actin microfilaments interact with the myosin molecules, causing the ring to contract. The contraction of this dividing cell's ring of microfilaments is like pulling a drawstring. The cleavage furrow deepens until the parent cell is pinched in two, protruding two completely separated cells, each with its own nucleus and own share of cytosol. Organelles and other subcellular structures. In plant cells, there's no cleavage furrow. Instead, in telophase, vesicles derived from the Golgi apparatus move along the microtubules to the middle of the cell, where they coalesce and produce a cell plate. Oh yeah, those little dots form. This is yeah, right. I forgot about this. They form a bunch of vesicles that have like cellulose in it or whatever. Alright, here we go. Binary fission in bacteria. Prokaryotes can undergo a type of reproduction in which the cell grows to roughly double its size and then divides to form two cells. The term binary fission, meaning division in half, refers to this process in the asexual reproduction of single-celled eukaryotes, such as amoeba. However, the process in eukaryotes involves mitosis, while that in prokaryotes does not. So prokaryotes do not do what I guess we call mitosis. In bacteria, most genes are carried on a single bacterial chromosome that consists of a circular DNA molecule and associated proteins. Although bacteria are smaller and simpler than eukaryotic cells, the challenge of replicating their genomes in an orderly fashion and distributing copies to equally to two daughter cells is still formidable. For example, when it is fully stretched out, the chromosome of the bacteria E. coli is about 500 times as long. I thought for us it was 250,000 times, so what are they even complaining about? For such a long chromosome to fit within the cell, it must be highly coiled and folded. In some bacteria, the process of cell division is initiated when the DNA of the bacterial chromosome begins to replicate at a specific place called the ORI. Wait, why is it called ORI? I guess it just starts for Ori. At the origin of replication, producing two origins. As the chromosome continues to replicate, one origin moves rapidly toward the opposite end of the cell. While the chromosome is replicating, the cell elongates. When replication is complete and the bacterium has reached about twice its initial size, proteins cause its plasma membrane to pinch inward, dividing the parent bacterial cell into two daughter cells. I gotta go for a walk. The neighbors, the plant, the gardeners are here, whatever. It's gonna be so loud. Alright, I'll be back in like 30. Okay. We are back. We are back. We are back. We are back. Wait, am I clipping? Test. No, I'm not clipping. I'm not clipping. Okay, so we're talking about binary fission. While the chromosome is replicating, the cell elongates. When replication is complete and the bacteria has reached about twice its initial size, proteins cause its plasma membrane to pinch inward, dividing the bacterial cell into two daughter cells. In this way, each cell inherits a complete genome. Using the techniques of modern DNA technology to tag the ories with molecules that fluoresce, researchers have directly observed the movement of bacterial chromosomes. This movement is reminiscent of the poleward movement of the centromere regions. During anaphase, but bacteria don't have visible mitotic spindles or microtubules. In most bacterial species studied, the two origins of replications ended up at opposite ends of the cell in some other very specific location, possibly anchored there by one or more proteins. How bacterial chromosomes move and how their specific location is established and maintained are active areas of research. Several proteins that play important roles have been identified. Polymerization of one protein resembling actin apparently functions. Okay, wait, hold on. Is this fission? No, this is plant cell. Okay. Wait, is it? Okay. So, they basically didn't explain it, but that's fine. The evolution of mitosis. Given that prokaryotes preceded eukaryotes by more than 1 billion years, we might hypothesize that mitosis evolved from simpler prokaryotic mechanisms of cell reproduction. The fact that some of the proteins involved in binary fission are related to eukaryotic proteins supports that hypothesis. As eukaryotes with nuclear envelopes and larger genomes evolved, the ancestral process of binary fission seen today in bacteria somehow gave rise to mitosis. Variations on cell division exist in different groups of organisms. These variant processes may be similar to mechanisms used by ancestral species and thus may resemble steps in the evolution of mitosis from a binary fission-like process presumably carried out by very early bacteria. Possible intermediate stages are suggested by two unusual types of nuclear division found today in certain unicellular eukaryotes: dinoflagellates and diatoms, and some yeasts. These two modes of nuclear division are thought to be cases where ancestral mechanisms have remained relatively unchanged over evolutionary time. In both types, the nuclear envelope remains intact in contrast to what happens in most eukaryotic cells. Keep in mind, however, that we can't observe cell division in cells of extinct species. This hypothesis usually only currently uses only currently existing species as some possible examples of intermediate mechanisms. The other mechanisms may have existed in species that have gone extinct, and we simply have no way of knowing. In dinoflagellates, uh, the chromosomes attached to the nuclear envelope. Oh, really? Now that's interesting. Which remains intact during cell division. That's super interesting. Microtubules pass through the nucleus inside cytoplasmic tunnels, reinforcing the spatial orientation of the nucleus, which then divides in a process reminiscent of bacterial binary fission. In diatoms and some yeast, in these two other groups of unicellular eukaryotes, the nuclear envelope also remains intact during cell division. In these organisms, the microtubules form a spindle within the nucleus. Microtubules separate the chromosomes, and the nucleus splits into two daughter nuclei. Pretty cool. I actually like the diagrams. Okay, wait, hold on. Okay, wait, hold on. 12.8. Where are we at in 12.8? 1, 2, 3, 4, 5, 6. Do they have answers? Are there answers to these? Are they in the back? Oh, fuck yeah. What are we on? We're on 12, right? 12.2. Oh, nice. Yeah, 6. How many chromatids? 12. Okay, cool. Comparison. Okay, yeah, we know the answer to that one. It's that in animal cells we have this whole spindle thing that pulls the sister chromatids apart, where in plants, how do they separate the genetic material in plants? Oh no, I think that there is a spindle in. Yeah, so it's the main thing is that plant cytokinesis is different, but they have a spindle so they can separate the material just fine. It's just that they can't. They don't do the thing where they elongate this way because they don't really. They just don't do that. Instead, they make a bunch of vesicles, and then those vesicles fuse. Whereas here, it's the draw. With animals, it's like the drawstring bag. It's like some actin or myosin just like pulls it shut. During which stages of the cell cycle does a chromosome consist of two identical chromatids? I would say G2. Is it G2? The end of S through the end of oh yeah, actually that's a more accurate answer. The end of metaphase, because it goes prophase, prometaphase, metaphase, Then what? Anaphase to telophase? There's 5. 1, 2, 3, 4, 5. Okay. So, yeah, so metaphase, they meet at the middle, and then anaphase is when they pull apart. That's all good. Compare the roles of tubulin and actin during eukaryotic cell division. That's a little more advanced than we needed to go. Yeah, 5. Let's see how they respond to 5. Oh, that's a lame answer. Okay. Ugh, I don't like it when it does that. Oh, nice. It saves the zoom. That's awesome. All right. The eukaryotic cell cycle is regulated by a molecular control system. Dude, we have so much bio. It's like this class is actually the amount of material to learn how life works. Who would have thought is like a lot. Like we got to learn how plants work. The timing and rate. Okay, wait. What is the concept 12.3? The eukaryotic cell cycle is regulated by a molecular control system. So nonspecific. The timing and rate of cell division in different parts of a plant or animal are crucial to normal growth, development, and maintenance. The frequency of cell division varies with the type of cell. The frequency of cell division varies with the type of cell. For example, human skin cells divide frequently throughout life, whereas liver cells maintain the ability to divide but keep it in reserve until an appropriate need arises, say to repair a wound. Some of the most specialized cells, such as fully formed nerve cells and muscle cells, do not divide at all in a mature human. Yeah, bro. What if you inject yourself with some iPSC stuff and got hella swole? You're like 50 and you inject yourself with some iPSC stuff and then I mean, or you could just inject stem cells maybe. I don't even know. These cell cycle differences result from regulation at the molecular level. The mechanisms of this regulation are of great interest not only to understand the life cycles of normal cells, but also to learn how cancer cells manage to escape the usual controls. The cell cycle control system. What controls the cell cycle? In the early 1970s, a variety of experiments led to the hypothesis that the cell cycle is driven by specific signaling molecules present in the cytoplasm. Some of the first strong evidence for this hypothesis came from experiments with mammalian cells grown in culture. In these experiments, 2 cells in different phases of the cell cycle were fused to form a single cell with 2 nuclei. In one of the original cells, if one of the original cells was in the S phase and the other was in G1, the G1 nucleus immediately entered the S phase. Ah, interesting. As those stimulated by signaling molecules present in the cytoplasm of the first cell. Similarly, if a cell undergoing mitosis was fused with another cell in any stage of its cell cycle, even G1, the second nucleus immediately entered mitosis. But what happened? So it always jumps to like the latest point, kind of. This is a cool experiment. Mammalian cell fusion, induction of premature chromosome condensation in interphase nuclei. Huh. The cell cycle is regulated at certain checkpoints by both internal and external signals. A checkpoint in the cell cycle is a control point where the stop and go ahead signals can regulate this cycle. 3 important checkpoints are found in the G1, G2, and M phases, which we will discuss shortly. To understand how cell cycle checkpoints work, we'll first identify some of the molecules that make up the cell cycle control system. The cell cycle clock, cyclins and cyclin-dependent kinases. Rhythmic fluctuations in the abundance and activity of the cell cycle control molecules pace the sequential events of the cell cycle. These regulatory molecules are mainly proteins of 2 types. What's a kinase? It cleaves a phosphate, catalyzes the transfer of phosphate groups from high energy phosphate donating molecules to specific substrates. Okay, so a kinase is more of it's like an ATP transfer shuttle or something like that. And cyclins, as you learned in chapter 11, protein kinases are enzymes that activate or inactivate other proteins by phosphorylating them. Oh, well, that's actually a little bit different than what I thought. Okay. Many of the kinases that drive the cell cycle are actually present at a constant concentration in the growing cell, but much of the time they're in an inactive form. To be active, such a kinase must be attached to a cyclin, a protein that gets its name from its cyclically fluctuating concentration in the cell. It's funny because I think, what is it called? Michael Elowitz's repressilator was based off the cell. I thought it was based off of circadian rhythms. Is it based off of the cell cycle? Anyways, I would love to look at, look at how cyclin proteins are regulated. Okay. Because of this requirement, these kinases are called cyclin-dependent kinases. CDKs. The activity of a CDK rises and falls with the changes in the concentration of its cyclin partner. Okay, figure shows the fluctuating activity of MPF, the cyclin-dependent cyclin CDK complex that was first discovered in frog eggs. I wonder how they did that experiment, how they found the coupling. The cyclin level rises during S and G2 and then falls abruptly during M. The initials MPF stands for maturation promoting factor, but we can think of MPF as the M phase promoting factor because it triggers the cell's passage into the M phase past the G2 checkpoint. When cyclins that accumulate during G2, and again, this is all mitosis, so like, I mean, that's why I'm like, man, I wish I could just learn molecular microbial biology, because it's like, bro, eukaryotes are so complicated, man. But no, this is actually pretty interesting. It's just a lot to remember, I think. When cyclins that accumulate during G2 associate with CDK, the resulting MPF complex is active. It phosphorylates a variety of proteins, initiating mitosis. MPF acts both directly as a kinase and indirectly by activate. Bro, this is too advanced. Bro, that's so scary. Okay, cool. All right, let's go through the review, and then I'm going to wrap this up. Ooh, and there's a test at the end of the chapter. That's dope. For selected answers, see Appendix A. That's cool. Summary of key concepts. Oh, that's great. I love when they have a lot of problems. Okay, unicellular organisms reproduce by cell division. Multicellular organisms depend on cell division for their development from a fertilized egg and growth and repair. DNA is the genetic material, or called the genome, made of chromosomes. Eukaryotes chromosomes consist of one DNA molecule associated with many proteins. Together, the complex of DNA chromatin, gametes and somatic, sister chromatids, centromeres, mitosis, cytokinesis. All this is pretty basic. Spindle, centrosomes, kinetochore. Oh wait, here we go. Cell cycle interphase. Yeah, this is the main thing to remember is prophase, prometa, meta, anatelo, cytokinesis. Okay, this is pretty cool. Okay, what's next? We have to do... Bro, we have so much. I'm gonna be studying like crazy this weekend, man. We need to do chapter 13, chapter 28. I need to do 1 and 26 still. 1, I don't know, I might just look at his slides. 26 I might read. I'll probably practice some more the thing that I want to do though is that, because like, let's see, what is 28? 28 protests? $35 for sure. 35 and 36. But what I'm wondering is, do we talk about $38? We probably do need to know $38. But he didn't put it in here. I guess what I want to know is, like, are we doing... Maybe this is the stuff that has... like alternation of generations and stuff like that. Let's look at the glossary. The book is so long that we can't even search it. Anyways, I guess we'll just rely on the notes for that. Oh, did I mess this up? Okay. Alright, let's just read some of these. Through a microscope, you can see a cell plate beginning to develop across the middle of a cell. It's most likely an animal cell in metaphase. Oh, well, animal cell, well... it could be this. Is it that? Oh, is that cytokinesis? Yeah, it is. It's not. That's not metaphase. I saw cell plate, and I got confused with the metaphase plate. Okay, vinblasting is... Okay, wait, we're going to skip that one. One different... Bro. The decline of MPF activity at the end of mitosis is due to... destruction of the decreased synthesis. Probably the degradation of cyclin. It's A. The destruction of the CDK. Alright, I'm not getting any of these. Okay, 5 is... Oh, wait. 5 is A. Okay, wait. I think I was... 4 is what? 4 is C. Oh, yeah, I was right. Which of the following occurs during S phase? Replication of the DNA. B. Cell A has half as much DNA as cell B, C, and D in a mitochondria. It's probably in G1, bro. I would say that actin is probably cleavage for a formation. Yeah, it's 8 is D. Alright, cool. We did the questions. Bro, I know about this guy, Francisco Mojica. I read that book about CRISPR. And I read that whole book on CRISPR. I still had no idea what the hell CRISPR was. It's like, why didn't someone just say it's a programmable cleavage thing? It's like, I don't know. Alright, that's it for cell cycle. I skipped the stuff on cancer and whatever, but